So we do this like a show and tell, where we share mushrooms that we have found in recent times from the greater New Jersey area, and either offer IDs, like tell people what we think they are, or seek help from each other and try to come to some sort of conclusion or consensus. Everyone's welcome to share, whether you're a beginner or an expert or anywhere in between. We take turns. So some people have emailed me already, and we also get like a running list going in the chat. So if you have mushrooms that you want to share from your own screen, just put your name in the chat, and I'll call on people in the order that they volunteer to share. And if somebody could volunteer to type the names into the chat box as we're going, it helps for people to follow along. So I saw Mintari says she has some mushrooms to share. Do you want to start, Mintari, or do you want to wait a little bit?
I will start. Last week I couldn't share because my computer in the setting, it didn't allow Zoom to look at my pictures on my desktop. So I think we fixed that. So I'm gonna try again now.
That sounds great. One more comment, I'm sorry, before we get going. I just asked everyone to please share the time. There's 18 of us here, and I know at least 7 of us so far have stuff to share. So we should really keep it to, 5 to 10 minutes tops. So everyone gets a chance. All right, cool. Go ahead, Mintari, whenever you're ready.
Okay, so I will not share all my mushrooms then. I will share only 5. We found a lot of them. You see it?
Yes.
Okay, so.
Although you may want to maximize your screen. We only see half of your desktop.
Oh, but do you see the mushroom?
We do.
Oh.
See it.
So that's good, no?
But it's half of the screen.
Half of,
Yeah.
half of the screen?
Yeah, if you could maximize it to fill your whole screen. There you go.
So this, I found them in, right here in Tenafly, Bergen County. According to iNaturalist, it's a kind of russula. I guess there's so many different kinds of russulas, if I'm not mistaken. I'm kind of a little, I'm hesitant to say things because there's so many mushroom experts in the group that I feel kind of, I don't want to say, things as if I know what I'm talking about. You know what I mean? So this is ridiculous.
You're not going to do any better than any of us on this one by saying that it's a russula.
Oh, so it's not.
Because that's as good as it's going to get with this one. Is it sticky?
Is it sticky? Yes.
Yeah, it's probably one of the fetid ones. Although the gills look kind of lacking in brown. It's probably one of the so-called fetid ones, unless if it was right after it rained.
I see.
Mintari, can you click on the picture and make it bigger, please? Thank you.
Okay, good.
Yeah, it does kind of look like that. It's got the bumpy edges on those, striations.
Right.
I think it's one of the fetid ones. There's like 4 or 5 possibilities. But I don't think we're going to be able to put a species name on this.
Okay.
Let's call it
russula.
Let's call it Grat and call it a day.
Oh, Igor, you got a proposal. Whoa, go for it.
Yeah, what the heck? What do I have to lose?
So Mintari, the reason why Dave's saying that is because russulas are so many of them, and they're so difficult to get the species. Even if you have a bunch of fresh ones in your hands and all the literature in a microscope, it's still difficult to pin them down.
So is it true that all russulas are edible?
No.
No? They're different.
Absolutely
colors.
not. Some of them will make you sick, and there are a few actually dangerous species. Very few, but some will make you sick.
Okay, there was a thing on Facebook this week. Somebody claimed that their dog had eaten a russula and died. Well, I checked with, what's his
Michael
name?
Buke.
Michael Buke, who's been keeping records for 30 years, and no animal, no person has ever died from a rustler. They have gotten sick, but not died.
Ah, interesting. Okay. But
Yeah,
you-
I'm pretty sure the dog did not provide a description of the mushroom. So there may have been russulas present, but the dog may have eaten something else like an anosogby or something like that.
Yeah. It was a puppy too. They eat everything.
They eat everything.
They eat everything. Okay. So I think my 5 minutes is up. I still have a lot of mushrooms to share, but maybe I'll wait till
Show us one or two more.
No, you've only used a couple of minutes. Go.
from, I'm looking at my clock, you know? Okay, if you want to show some more, I can do that.
Mintari, do you have the gray brownish cap that you, Russula, that you found at Kittatinny?
And Kitatini?
Yes, do you have
Oh,
that?
yes, yes. Oh, wait,
Why don't you show that? Because I identified it.
right.
No.
Is it this one? Right?
No, that's not it. It was.
That's not it. This is also Rusula, am I correct?
Same as the last one. Go to another one.
Okay, go to another one.
Gray. Doesn't have a picture.
Oh, it's okay.
She doesn't have a picture, I don't think.
Never mind.
I might not have taken a picture of it.
I took a bunch of pictures of it, but
Oh,
I'm
it's
not.
this one.
Yeah, but he doesn't show his pictures in public. He's shy.
So, okay, can I share this one here? Because I'm curious. This one here. So this is actually, they were in Tenafly, in Bergen County. They were on like a stump that a big tree that was cut. Look at that. So what is that?
Epidotus.
No.
Yeah, that'd be my first guess. Gills look kind of dark.
Yeah, that's my guess, but they don't usually cluster like that, do they?
Yeah, they cluster like that. The one that's got me worried is lower left that seems to have a pretty thick, almost centrally located stipe with a split on the lower left leading toward the stipe. That's a Hohenhabilia type of trait. But the dark gills point toward Crepidotus.
So, all right. So that's definitely not edible.
Oh, no.
Either way, not edible.
As you can see, I'm into edible mushrooms.
Yeah, people really need to know what it is before you're going to eat it.
Oh, yes. So, like what I said to Igor, in Tenafly, in Bergen County, what happened to the pictures? Because I took a picture of the whole field full of chanterelles. Oh, it didn't get uploaded. But this is definitely Amanita. Look at that. Right? It's one of the Amanitas. Look
Probably need to see the bottom of the stalk though, but the skirt-like ring seen on two of the small ones in the lower right, that certainly looks like Amanita to me.
right.
Maybe Amanita submaculata, but you have to dig it up.
Oh, okay. You know, I assume that all mushrooms that have a skirt like this, a ring, they're all like poisonous, but that's not true with this one. I want to show you this one.
Yeah, that's definitely not true. Think of a portabella.
Oh, portabella.
Portabella's got a big old annulus on it.
Yeah. Okay. I think my time is up. Igor, you have to wait for all my bullets next time because I have a
Sorry?
Yes.
But you don't get poisoned by touching them.
By touching this?
Even if it is an amanita, if it is poisonous, you don't get poisoned by touching it.
No, no,
Okay,
right?
You know that, yeah?
Yes, I do.
Oh, okay.
I
Okay.
learned it from this group, by the way.
Okay.
Okay, I think my time is up.
Show us a bully.
Oh, show some bullets? Okay.
Don't
By the
worry
way,
about the time. Look,
I don't think this is. I don't think this is a Manita submaculata. The that one has an apical ring. The ring here is not apical. It's in the middle of the stipe.
Okay, okay. Let's look at a bully.
Bullets. Okay, so hold
Something
on.
unusual.
Something unusual?
Yes.
Unusual. I don't know. You know, they're all unusual to me.
I was gonna say that.
Igor, everything is usual for you, but everything for me is unusual. You know,
That's a good, that's a fair
it's all
statement.
relative, right? But look at this. It's supposed to be porcini.
This is easy.
Easy.
All
Oh,
right, an edible one.
oh, not edible one. Okay. So
This
hold
is
on.
boring. Let's move on.
Boring. Okay, so hold on.
What is
No,
it?
it's Boletus separans.
Severance,
It's
Yes,
a good
correct.
edible as long as it's not a
No.
insect
Whoa.
condominium.
Go up. Go
Up.
up. Yeah, this one with the yellow pores.
You mean which one? This?
No,
No.
drop right above it.
This?
No. Yeah, this one. Yeah.
Above the separans, above the one you just. There we go. That's interesting.
Why are you interested in this one?
Because we don't know what it is.
Oh,
Because I don't know what this is. Yeah.
it's supposed to be bullet. A bullet.
Well, obviously it is a bullet.
Come on, Igor.
Can we enlarge this one, please?
Huh?
Can we enlarge it, make it bigger?
There.
Okay. And scroll down so I can see the stipe
Scroll
the other
down.
way.
Okay, hold on. There.
Hmm.
Yeah,
Now
that's a
it got
tricky
you.
one.
Got you, Igor.
Yeah. The pictures don't do it justice. Let's move on.
Well, hold on. Do you have more details about it? Like, does it have a smell? Does it have a sticky texture? Anything else?
No blue. This one will not blue. I'm pretty sure of that.
How about Hortiboletus harrisonii?
No, it's not hortiboletus.
You don't think it's Hortiboletus? It can't be Hortiboletus? Why not?
Just doesn't look like it.
My friend ate it, by the way, and she's still alive.
Well,
You should have saved this for me, you know, would have sequenced it for you, but forget about eating boletes. That's verboten.
Well, when we were doing the foray last weekend, we found poisonous bullet. Oh, no, bitter bullet.
Okay.
I can't
you're not
believe you're
interested
saying
because
that, Igor.
it's
I know.
Don't eat
Dave,
both.
other than hortiboletus, do you have any ideas what this could be?
Oh, this one?
Oh God, Xerocomus. I don't know. That's a trick. It's a tricky one.
It's a tricky
Maybe
one.
like something like Boletus rudii. I don't know. I'm just
It's not
grasping at
rudii. It's not rudii, but
not
it
rudii.
is, you know, it's something from the Xerocomoid series, you know, Oreoboletus, that kind of thing.
What
Yeah.
about the ornithypes?
No, stipe is wrong. Need to have yellow stipe with
reticulation.
coarse
Yeah.
reticulation.
Yeah.
Yeah.
Miss Hari, did you say that your
The cap
friend
color's
ate
wrong
that?
too for that.
I couldn't hear. What was it, Marisol?
Oh, that was me. I'm asking, did you say that your
Your
friend-
sound is pretty bad, Lauren.
I'm like talking right into the computer.
Lauren,
Lauren.
I'll type it.
Okay. It sounds cocked.
I have another proposal. Ha
Yeah.
ha. I think this might be another Boletus separans. It's just mature and the stalk is a little bit overexposed, so you don't see any ornamentation on it. But look at the cuticle. It's got the bumpy, slightly mauve sort of color. What do you think, Igor?
No.
I've got another
I
one.
know, I mean, I thought before it looked more porcini-like, but I didn't see any reticulation. And usually separans has at least something at the apex, you know, a little bit of that white netting.
Yeah, well, sometimes separans has virtually nothing.
I know, I found one
yeah.
actually recently. I'll probably posted.
I think this might be another separans. I'm guessing this is a separans.
That's a pretty bright yellow.
The only thing that throws me off is
surface.
the pores might be a little bit green for that species.
Yeah,
Yeah.
usually I was thinking separans, like I see them when they turn yellow pores, but that's usually kind of like a dingy.
Yeah, the pore color is a little bit off for that species, but what you can see of the cuticle looks like separans. But this is today's mystery bully. We're
Mhm.
we may as well move on.
Yes, okay.
Sorry, I shouldn't have said anything else.
All right, I think I'm gonna stop, let other people share.
All right, thanks, Matari.
Oh, you're welcome. Stop sharing. Okay, let's stop sharing right here.
Okey-doke. I'm going to go ahead and share because I do have some. Okay, I have observations from 5 people, beginning with Susan. She was the first one to email me today.
Yeah, I've had kind of a busy day and a bunch of leads from over the weekend and even a couple of things I just saw a little while ago, but they're not up here. Right, so I think this is the true Chalicoporus rubinellus. That color is pretty true. I'm sorry the picture isn't better, but they were absolutely gorgeous. And there was like 30 of them under a hemlock tree down in Blue Mountain Lake, which is about an hour south of me. I was, well, there for sort of a mushroom walk, sort of, with somebody. Aren't those pretty? Okay, so
Gorgeous.
so the next one.
Are they bright yellow right along the edge there? So,
Yeah, it's kind of a yellowy, it's a yellowy white color. Yeah.
okay.
And very pink pores, pink on the stem.
Mhm.
But the rosy pinky cap is almost kind of velvety. Velvety and smooth at the same time.
It's
Interesting.
like a peach.
Yeah, sort
said
of.
Yeah, like a peach.
Yeah, but redder, much redder. I've seen them there before. I just don't get down to that site that often, but we've had a lot of rain up here. So there are all kinds of mushrooms coming up everywhere. Okay, so I think I have a couple. Yeah, okay. Now this everybody's going to guess, oh yes, Swillus americanus. It is not Swillus americanus, it's Swillus subaureus. The differences in the pores are much oranger. And this picture is not showing the color as true as it is. The color on the cap is pretty much a little oranger than Swillus americanus, but the pore surface underneath is much oranger. And note how tight the pores are. This is at
Susan, did you find that near oak?
No, we have no oaks up here.
That's what I thought. I thought subaureus was primarily associated with oak, but I know there was a study not too long ago to challenge that hypothesis.
No, they think it's with aspen, and there was aspen around there.
Oh, okay.
It's from the same spot about 4 years ago, and actually physically gave them to Ritus Vigilis down at the NAMA foray down in Virginia. And he was very excited because I included a leaf, and he had evidence from a mycologist somewhere in the country who was trying to establish if this mushroom was mycorrhizal with aspen, which is unusual for Swillus because most of them are with pines and conifers. So anyway.
Yeah, that's what makes the Swillus subaureus that I find around here unusual, is that it's usually in an area with oak.
But it can also grow with hemlock. I know that.
Really?
Oh, okay.
It's quite promiscuous, I think.
Oh yeah, now that's kind of interesting, isn't it?
Yeah,
See
just
both hemlock needles and an aspen leaf in this picture.
Yeah, I put that aspen leaf there.
Okay.
I don't remember that there was any hemlocks in that spot, but it could be. But there are balsam fir, red spruce, eastern white pine, a couple other things. So anyway, I'm always excited when I see this. And it does come up in the exact same spot every year, at least 1 or 2, but the other day there was quite a few.
Do you do anything with it, Susan?
This particular collection, no, other than photograph it. Like I said, 4 years ago, I actually physically gave it to Regis at the foray at, gosh, I can't remember where. It was at Nama foray down in Virginia, hot as old get out.
And this wouldn't produce like a good color for you for dyeing?
No, Swillis, to my experience, aren't even worth bothering with. But then again, if I want yellow or orange or a nice dark green, I would look for Phaeola schweinitzii, which is a polypore, which should be out now, but I just haven't stopped and looked for it because I have 3 boxes behind me stacked, dried.
Say the name again. I want to see if I have that.
Oh,
I've got one, Bianca. I'll show it later.
Perfect. Thank you.
Oh, for the dyeing? Mhm. Okay, next one, Luke, I forget what. Oh, now this was also down in Blue Mountain Lake, which is about an hour south of me. I called that's what the one, Igor's put in the name of the one we were just looking at. This one I have been calling.
Porphyrolis.
Yeah, Porphyrolis is a new genus for this. It used to be Tolopolis.
I put it in as Tylopolis sordidus. Oh, good.
Yeah.
I got the species.
Yeah.
Right. All right, so you're calling that Porphyrolis?
Porphyrolis sordidus.
Yeah, there was a paper by Michael Kuo and Beatrice Ortiz Santana from 2020, and they transferred this to Porphyrolis. It's pretty clear.
I do not see it that often. I'm just trying to copy down your name here. And then there's the color of the blue from this gray brown color is pretty distinctive. So I knew it was pretty weird, or at least weird for me. And too bad it was a little old.
Do you get the red staining on the flesh as well, blue and red?
Oh, I didn't notice that. This was so over the hill, I did not keep it. The next picture shows the nice kind of medium brown, smooth color on the cap, but it also was kind of half eaten away. And
You can see the red on the pores, a mixture of blue and red. That's what it does.
over here on the left.
And on the previous picture as well, yes.
Okay, okay.
Yeah, I found a few of these last week. I did not include in my submissions because there are just so many mushrooms.
So what do we think it's mycorrhizal with?
I find it with oak.
So do I.
Up there, it's something else.
It is possible at this spot that there could have been oaks, but I did not see any, but it's far enough south of me that it changes habitat below Tupper Lake a little bit. And I know in that area I have seen oaks. I just wasn't, really focusing on that particular walk. But there's a lot of beech in there too. And almost no conifers.
The spot where I find it also has shagbark hickory, but you probably don't have any of that up there at all.
I don't think so. Okay.
I think there's been some confusion about Tolopolis sordidus. There could be another species floating around because I remember I read it in Ernst Both's Compendium of Boletes, which is a very useful reference, even though it doesn't have any pictures. Carl B. Wolf, who was very active, who actually wrote a book about Porphyrolis back in the 80s, he discovered a species that looked like sordidus in New Jersey, in upstate New Jersey, and he called it, I already forget the name, but there's something probably else out there. So
I remember C.B. Wolf.
Oh yes, C.B. Wolf.
C.B. Wolf.
Yep.
Montalto.
He's still alive, I think, but he is getting closer to 80.
I think he moved to Florida. Didn't he have his collection at Montalto and somebody moved it?
Yes.
State?
Possibly. I never met the gentleman. I would have loved to, but he's retired now.
He was at the 1982 Nama foray in East Stroudsburg. And if we have time, I have another email with 4 more mushrooms.
Yeah, let's see them.
Yeah, one difference between this Porphyrolis and the ones I find here, which exhibit the same kind of staining, the cap surface on the ones that I find is generally darker, and it breaks up and it has a lot of cracks on it. not that long after
This-
it pops.
In my area, every mushroom has been almost completely waterlogged, but I would suspect that in drier conditions that would happen with more boletes. Okay, so this is one of my Inocybe collections. It's not been identified yet, but it's on its way to University of Tennessee to Brandon Matheny, who I've sort of had a busy day communicating with about several collections I made of Inocypes, which I usually ignore, but any of you who were on that Zoom program about a month ago, he sort of charged us to look at Inocypes. So I'm trying to do that. And I'm very excited because he's coming up here with his lab team for a week in August. So that's really exciting. But this is unusual to be able to find this many Inocypes in all stages at one time that are not, you know, disintegrating or in good enough condition to photograph.
Susan, what does it mean that they're going to be coming up with lab team to here? Does that mean they're going to be in the area trying to study the mushrooms in this?
Yes.
Okay.
Brandon Matheny is trying to make a monograph of worldwide of Inocybe, and they're his pet peeve is Inocypes, which God bless him for looking at them. But he's one of our top DNA guys, and he replaced Ron Peterson at the University of Tennessee as a mycologist there. And he has a lab of, I don't know how many students, I don't know how many he's bringing up, but they're staying over on Lake Placid. So I'm only 7 miles from Lake Placid, so I hope to connect with him. I told him where the grocery stores are and some of the other things.
Susan, do you know when in August? Because I'm going to
Yeah.
be in the Adirondacks in early August.
Second week.
I'll be gone. I'll be gone then.
Yeah, I can't believe they're actually going to spend a whole week. I'm not sure why he chose that, other than it could be maybe some of the Peck original type collections were from that area. The plants of North Elba would be my guess.
Well, they like to get collections that span geographic diversity.
Right,
Yeah.
right.
So, yeah.
Well, like I said, if you saw that Zoom program, he sort of charged some of us New York mycologists to look for Inocypes. I can recognize the genus, but I don't get any further than that.
Can you tell us
Actually,
about-
there's like 4 genera now. It's been
Oh, yeah.
split.
He
Yeah.
went over the whole thing in this program. And
Yeah. Yeah, I saw it. It was
Yeah,
a
it
good
was quite
program. He
good.
did a nice job.
So
Can
the
you give a high level overview of what an Inocybe would be compared to, for example, an Amanita?
They're small. These are no more than about 2 inches tall, maybe 2.5 inches. They usually, when they open out their umbrella, which is like the one here on the middle and the left, they have that kind of distinct umbo kind of nipple kind of structure. In this case, the stems are white, but not always. They're under the gills are kind of a non-distinct gray browns. Foreprint is kind of a light brown to a dark brown. They have very interesting spores if you're into microscope work. And the other thing which you can see in the one, just the dead center sort of, they look like they have had their hair combed out from the center umbo out to the edge of the gills. And the whole thing, like I said, is no bigger than about 2 inches across when it's opened out. They're mycorrhizal, and they can be plentiful after a rain. And they often smell really bad. Other than that, they're boring little mushrooms. But
Thank you, Susan.
he loves them, absolutely loves them.
Mostly poisonous.
Yeah, very poisonous. Yeah, that's true.
Did anybody see this week some of those Inocypes from Ohio, I think, that Alan Rockefeller was posting that that he says has psilocybin in them?
Oh, that might be true. Yeah.
There's a few species that have psychoactive components.
Yeah, they were really, he put some really beautiful photographs. If anybody's on Facebook, and if you just dig around looking at Alan Rockefeller with his Inocypes this week, some amazing micro photography he did of them too.
Yeah, I see in the chat that Inocybe, Brandon has divided up Inocybe into several, I would call them subgenera, but you know, he's the one that's doing the work on it. And he, as I remember, there was about 5 different subgenera or genera that he's created, which will be several years or not soon for us down in the cheap seats, as it were, before we get that. Anyway,
If you use a microscope, you can probably learn these new genera.
yes,
Because and the new thing is like it's like I guess the word is family in Osabaceae,
yeah,
and Osabe is still a genus.
yes,
It's the one with I believe Osabe are the ones that have the lots of pleurocystidia with that are apically encrusted, and the nodulose or stellate spores for the most part, but then I think he said that some of them just have the ellipsoid or bean shaped spores. But it's it looks like most of the sectioning, the partitioning into these new genera is at least partially based on microscopic traits, which you might say, oh, geez, microscopic traits. Well, that's better than just having DNA do all the talking, because at least you can look through a microscope and see something, right, without having to send it to a lab. So
they do have interesting spores.
yeah, some of them do and some of them don't. Some of them just have these big bean-shaped spores. But some of them have these like spores that look like, those World War II aquatic mines that they would, they look like that. That some of the
Like
spores are
minesweeper.
really pretty cool looking. True.
Yeah, that's true. It's true. Anyway, I saw a lot of things over this past weekend. Luke, the next one, is it a Leptonia? Yeah, this is a Leptonia. I've called it Cerulata. That's a kind of a cluster of species, but what you can just about see on the one that's kind of lower center is that the gill edges are kind of bluish, like the gap. They're little, tiny. They're pink spored, which is Timberoni's bailiwick. And they are also very small. I recognize this genus, Leptonia, by the belly button, kind of on the center of the cap, and also the slightly scaly look to the cap texture, again, sort of radiating out from the center cap. Small, all different colors, or at least yellows, light browns, a lot of them are light browns, but they're interesting in that they have pink spores. And what I find up here in the Adirondacks is when they come out, they like 6 or 8 species will come out at the same time, and they'll be out for a few days or a week or 2, and then boop, they're gone. Don't see them again. But this
I
is
find a lot of these when I'm in the Adirondacks, and like you said, Susan, different kinds. And another trait that's generally associated with these Leptonias are the jagged gill edges.
yeah, slightly jagged, and in this case, you can see the fact that it's Chylocystia are blue along the edge of the
right.
really cool.
Right.
Really
so
cool.
we would call that marginate edges, right?
Marginate edges, yeah.
The edges are a different color.
Oh, that's real. That's Chylocystia.
I'm pretty sure that's right.
Because that's a bit unusual in the Entomomaceae. Because a lot of Entomomas have no cystidia at all.
I'm assuming that's what it is, but
I'm going to check that out. That's an interesting suggestion.
Timberoni might spell that out.
Yeah, I have Baroni's book. I'll look in it. But you might be right about that. That would be a reasonable, very sort of Occam's razor sort of explanation for why the gills look like that.
Spathulariopsis velutina. I learned it as Spathularia velutina, but now it's got Eopsias on the end of the genus. I see these kind of a lot up here in the Adirondacks. These are about 50 feet out the back door from my house on a well-rotted log. They're really cute. They're only about an inch, maybe an inch and a half tall, and maybe a half an inch across. But they're just these funny little fans that form their spores all, I think, all on both sides of that upper surface of the fan. Cute. Wood rotting, I think, fungi. Oh yeah, and the next one is, again, something that I saw quite a few of this past weekend, the Hemilexinum subglabripes, or I learned it as Boletus subglabripes. Very pretty. This picture, the yellow of the pores is not quite showing up as beautiful as it is, but very small, tiny pores, and a slight pink flush down the stem. And I've eaten this and it's pretty good. And when they're out, there's a lot of them out.
This looks very similar to the other one that was shown. Do you think so?
Yeah, the stem is quite different on the other thing. Plus the gills, the pores on the other one were much yellower.
This is a boreal taxon. We don't find this in New Jersey. I think it's mostly birch associated.
Oh, that would make sense. Yeah, there would have been birches there. Yeah. Yeah. And I think that's it, right, Luke?
Looks that way.
Dave, I don't think you find this thing in your area, right?
Oh, I'm just getting back. I took a break. Was that subglabripes?
Yes.
Yeah.
Yeah. Rarely. I found it in Binghamton when I was a student at the university in Binghamton, but not that commonly. Rarely down here. I find it in the Adirondacks and in Vermont. It's very common in the mountainous areas.
It's a boreal species. It doesn't
Yeah.
grow south
Not a
of.
bad edible, actually.
Yeah, I've eaten it. It's okay if you get them young enough before the bugs get into it.
Yeah. Yeah.
Cool. Well, thank you, Susan.
And I'm not much of an eater of fungi, don't forget.
Okey doke. Marisol, you are up.
Yep. All right. Okay, I found these Gymnopilus. I don't really know too much about it. Maybe like 7 centimeters white cap. This one was a little deformed. You can see on the other pictures. The spore print is rusty. This is a younger one, but on the top, and there was a bunch of them tiny growing together on deciduous wood. Super tiny, they were babies. Those are the gills, very crowded. I got the spore print, and everything in water is yellow. It's beautiful. So here you can see the basidia and capitate cystidia. I don't know the names. I can't put it in my head. So you can see that the cystidia is empty, and or I don't know if that will be, no, because there are no stigmata. Yeah. So that's no empty basidia. The basidia is yellow. The cystidia is clear. It's a little more extended than the basidia layer.
I always have a hard time with that, but my microscopes aren't that good, really. But the basidioles versus capitate cystidia really is confusing.
No, but they are bigger than the basidia, and they don't have any remnants of stigmata. I see empty.
Yeah, but the basidioles are these like sterile basidia. So I find them like confusing.
Oh, I didn't know.
Yeah, I always thought, you know what? Check me on that. I might be wrong.
Okay.
Did you measure spores for this?
Yeah, I did. And I noticed that what I am calling capitate cystidia has, I put a yellow arrow at the lower right, showing the septa on the clear structure. I couldn't get a more clear photo. That's what I got. But I love the color. Those are the spores. I got the measurements.
If you mount Gymnopolis spores in melters, you should get a dextrinoid reaction.
So they will be like dark?
Yeah, they'll get a little bit purple. It's not, in my experience, it's not a real strong reaction.
Okay, okay.
But yeah, they get a little bit purplish.
But what's the difference between Gymnopus and Gymnopilus? I want to know that because I don't know.
I'm sorry, what's the difference in what?
Gymnopus and Gymnopilus.
Oh, well, Gymnopolis are these rusty brown spored mushrooms that grow directly on wood.
See,
and Gymnopolis are these pale spored mushrooms that grow on forest debris, sometimes wood. But the sounds of the names of the genera are just merely coincidental. They're not related.
Yeah, so this one is a Gymnopus?
Gymnopolis.
Ah, that's the name I put in there.
Yeah, Gymnopolis.
Okay, so I was right about the Gymnopolis, that's all. But I don't know the species, no clue.
If you got spore measurements, we can probably narrow it down to 2.
Oh, okay. I got them.
And if you know hardwood, I think that would point in one direction.
It is
but I forget which way.
Where I was, there were no conifers. Only.
Yeah, are the spores really small, or are they more or less in the 7 to 8?
It's 7.4 by 4.9.
Yeah, that's the typical for like Sapinius or penetrans.
Oh, so we get.
There's 2 species, Sapinius, penetrans.
Penetrans,
and one of them favors conifer and one of them favors hardwood, but I believe it is suspected there's some overlap there.
Okay.
Yeah, so it becomes pretty tricky. There's another one, Bellillulus, but the spores are very small. I don't think they get much bigger than 5.5 microns.
What's the name? The last one?
Bellillulus, to the best of my memory.
Can you spell?
Well, I'm not even sure I'm saying it right.
Idea?
But it starts with B. B-E. B-E-L. I'm pretty sure about the B-E-L.
Okay. But I tell you something. I remember that in Franklin Parker, we find Gymnopilus penetrans all the time.
Yeah, penetrans is pretty. Penetrans and Sapinius are pretty common.
Yeah, but
the Bellillulus seems a little bit more uncommon.
Okay. So
and once you measure the spores, you can pretty much, tell if it's Bellillulus or not.
Okay.
Because the spores will be really small for Bellillulus, and there might be some other species that have small spores, but I don't know of any other names.
Okay. Thanks. I think there is one more photo. Look.
Yeah, those look like Gymnopolis spores. Alan Rockefeller once told me he thinks of Gymnopolis spores as being shaped like mangoes, which is a pretty good description.
Kind of, yeah. Okay. Okay, thank you. Okay, this is really bad because you cannot see anything in there, only these little white spots. It's a resupinate polyp, you can't see anything. I took the photo at home when the thing was all dry. Somebody gave it to me in the foray in Horseshoe Bend, and I was so excited because I found out the species. It's a Trechispora, it's a resupinate polyp, I said that. And one of the characteristics of the Trechispora, there are 2 that are very close. One is Trechispora mollusca and one is Trechispora hemiocystis, which has very particular globose structures that I will show you at the end. So this is called ampullate clamps. It's like inflated at the place of the septa and the clamp. Yeah, but this one was a little odd. It has 2 legs at the bottom. And you see all other weird ones. Oh, I pointed to 3. Okay, here is, I pointed to 4 of them. Immature basidia at the left and an empty basidium at the right, and the spores are growing. I love that the basidium is empty because it gives all it is matter to the spores, 4 spores. They're super tiny. And then more of the ampullate clams, and this one was a weirdo. It has 2, and you can see one on the right in the center that is more regular. And that weird thing on the right is like an extra. I don't know what it is. All right, thank you. And those are the spores are ornamented. They're super tiny, but it's one of the other characteristics of Trachyspora, the ornamented with spines spores. I have more photos. Yeah, it even have very odd-shaped ampullate clams. Oh, I put odd-shaped versus regular ampullate clam. Basidium that I found floating, and one spore in the left. And this is a photo that I took from somebody else online to show you how the resupinate polypore looks. It's very delicate. It's very small on rotten deciduous wood. And I have the microscopic features for the other Trachyspora that is very close to this one. Can you pass to the last one, please? It has these globose, huge structures, sphirocysts. So that's what makes the difference between the 2 Trachysporas that are resupinate, that are polypores, resupinate polypores. Thank you.
Marisol, you said these were spirocysts?
Yes.
So does this put this in Russula Elias?
I don't know. That I don't know. This is a hymeno. That's what it gives. All I know is that these elements give the name to the species, Trachyspora hymenocystis. Cysts. That's all I know. But I don't know more than that. All right.
Cool.
Mm-hmm. Okay, somebody found this one in our Kitatini State Park and gave it to, I took it to work on the micro because I could not recognize it. It was very small, 1.3 centimeters for the one on the lower part. And it looks like a carmine red kind of Scutellinia, but it doesn't have any hairs. And when I turned it on the other side, it was wrinkled. It has wrinkled surface and then nothing, no food. But if you look, the one on the top, I took home the whole, everything there, and then I watered the one on the top. And when I look underneath, it has a foot, a whitish foot. I did the micro. Can you please pass? Oh, another photo. You can see the wrinkled surface underneath. Mostly, I never saw anything like that. The color is pretty special, and I work on the micro, and the spores are not ornamented, kind of like a oval shape, kind of. Not ornamented, and the very special about the paraphyses, which are the infertile parts that are between the asci, they have red pigments that you can see in one of the photos. Can you please pass? I did it like that so you could see the pigments. It's really beautiful. This is in water. Mm-hmm.
Yeah, nice job on that.
And those are more spores, but this is the problem. It looks like Sarcosifa occidentalis, but it has no hairs, no kind of any hair underneath, and the shape and size of the spores doesn't match. And also it doesn't have a sheath. They say there that some of these Sarcosifa have a sheath. I could not find that. I did it again and nothing. I could not find it. But I was reading about the Sarcosifa, and it seems to be like a group. So there are other ones that don't have the sheath and don't have are not as big, but I couldn't get to the species.
Yeah, but the occidentalis is the only one with a stipe.
See, that's why I call it occidentalis.
Yeah, I think you're probably right. I just made a couple of posts on MO during the last month that I call by the same name. I'll send you an email. With
Okay,
some
do,
links.
please.
Mm.
All right. Or I don't know if the spores were not mature. I have no idea. That's
Yeah, I
okay.
think my spore measurements may have been a little bit different than what was.
Mm.
I don't quite remember, but we can compare notes.
Oh, okay. Mine don't coincide with the sizes they give. The sizes are way too big. But it was very interesting little tiny thing on a little stick. Right.
Yeah, they grow on sticks, little sticks.
And I thought, according to what I read, they start in spring. And apparently they grow in spring, but look, it's July and it's so hot and we found it. Somebody found it.
I think I just saw one the other day. They grow on my property. So I see a lot of them. And I think I just saw one the other day, but I didn't. I was kind of too tired from bending over picking mushrooms, and I didn't actually get it. But it looked like one of these. So I think they're still coming out here in my spot.
Yeah, I was surprised because they said in spring. Okay.
To see them years ago in New Jersey. I would have said late spring.
What I was reading, yeah, it said spring. So, okay, this one was such an adorable little tiny LBM, and it was found as I am showing to you, and I call it like a Japanese dish. It has two chopsticks under the wood where the mushroom was growing. It's like on bark with other pieces on the top, and then the mushroom was growing in there. Can you show other photos? So the gills are pink, and I thought it was a Pluteus. It doesn't look to me like anything else. But I don't know if all Pluteus have this shape of cystidia that I call Greek base, like with incrustations on the top, because this one didn't have it. So I did the spore. This is late in the afternoon. It was so humid and so dry that it just fell by itself. All right. And I found this cystidia with septa at the base, and there is something really interesting. Oh, maybe it's another one. Okay, maybe I need to see more photos because I work in so many things that I am confused now. Okay, basidia is kind of fat, like in the center, and it has 4 stigmata. You can see the tips showing in there.
Nice.
Okay, and then those are the spores. I don't remember. I don't really know Pluteus spores yet.
Yeah, they all look the same.
They do like that? Look like that?
Yeah, pretty much.
Oh, and then
Subglobose, globose,
and that's why.
broadly ellipsoid, and many of the species exhibit range through all 3 of those categories. It's all about the cystidium. And the cystidium you showed, and the size of the fruit body too. This is not Cervinus. This is not a deer mushroom. It's too small, the stalk is too thin. There's a lot of interesting Pluteus species out there, and Justo, Alfredo Justo he's still studying these, and I would send him more material, but he's up in Canada now, and it costs like $30 to send a mushroom up there.
Oh no, I found a way to
send. So because I sent him a lot of stuff, and but that cystidium that you showed does not look anything like.
That's what I mean. Okay.
From Cervinus, or even from Pluteus section Pluteus. Looks like something else.
See, I didn't call it Cervinus, but I wanted to tell you something that I saw.
Yeah, you called it a deer mushroom, though, and that's the name.
No, not me. That's what they put in iNaturalist. I call it Pluteus.
Yeah, well, okay, fine, iNaturalist.
Yeah.
Deer mushroom is Pluteus cervinus group.
I call it Pluteus. I didn't call it anything else. Because I don't
ignore the iNaturalist thing there.
Yeah, I don't do it like that. I don't know how to show that. I don't do it with the common names. Okay, but I wanted to tell you something really cool that I saw here. So I saw the cystidia in different stages of development, and it starts kind of rounded, then it gets a tip, and then it develops a shoot. I could not focus that end, but you can see it with the yellow arrows. So that is the mature one. That's the
Oh, so it has like these projections.
yes,
Oh,
one.
that's pretty darn interesting. I've never,
Yes.
maybe my scope's not good enough to see something like that,
Yeah.
but that's,
So yeah,
but these
here-
are not section Pluteus. These are something different.
okay.
But they are genus Pluteus.
Oh, okay. And look at in here. So on the lower part of the cystidia, there is a little ball.
Yeah, wow,
So
very
before
nice.
it is starting to develop, to mature. If you look at the one on the lower right, it's a little bigger. And then at the end, it's a shoot. It's a long, I have all the measurements and everything on the paper. So
Wow.
I never saw anything like that. I was like, holy moly.
You know what, Marisol? I'm going to send you contact information
I
for
will send it to
Alfredo
him.
Justa.
I will send it to
And
him.
he will have something to say about
Okay,
this.
that's really cool.
Yeah, this is pretty interesting.
And here I found, I could not focus better than that. So I wanted to show you the basidia with the forestering matter in the left and the basidium in the right with the forest spores in the front. That was so cool. Like you seen this thing almost in an angle, but still it was fun. I think that I have more photos. I was fascinated with the cystidia.
You know, Maricel, Michael Kuo, mushroom expert.
Yeah.
He's got a pretty detailed Pluteus key on there.
I'll go there, okay.
It might be a little dated, but I know it's pretty extensive.
All right. And this thing was like maybe not even 2 inches tall. It was a tiny little guy. Look at the base of the cystidia. There are 2 coming from one septa thing in there. And there was plenty of them. This is like 100x. You can see how many of them were. And I think they were on the cap too. The cystidia was on the gills and on the cap. The cap looked sugary from the cystidia.
Pileocystidia.
I just can't put those names in my head. I had to
that's the name for the ones on the cap. Pileocystidia.
Wait, I have to be good. Okay, Caelocystidia is for the cap.
P-I-L.
Oh, Pileocystidia.
Yeah, P-I-L. I think it's
Pileo. Yeah, I know how to do it.
O. P-I-L-E-O cystidia.
Yeah,
I
and
think.
the caulo.
Those are the ones on the cap.
And on the gills is caulo.
Caulo on the gill edges.
Mm-hmm.
I'm sorry. No, I'm sorry. Caulo. I'm sorry. No, that's wrong. Caulo is on the stipe. Chilo. C-H-E-I.
I know the words.
That's on the gill edges. Pleuro is on the gill faces.
Pleuro.
Maricel, do you have,
Pleuro. P-L-E-U-R-O.
Yeah.
That's on the gill faces.
Yeah.
And,
I'm going to check on
Pileo. No, sorry, Pileo is on top.
You can
Caulo, C-A-U-L-O is on the stipe.
You can find all those names on like Mushroom Expert too.
Yeah, it's on all that stuff's on Mushroom Expert, right? Yep.
Okay. Maricel, do you not have the Largent series book of how to
see about the microscopic
features?
Yeah. I'm going to learn those things. Thank you.
Yeah, it's in the volume 3. Microscopic features.
In the place where I found these little Pluteos, I found this blurry crust, and it was a Botryobastidium conspersum. I have found it so many times. It's cottony. It has a cottony aspect, kind of white grayish, and I love the microscopy. It's the anamorph of a crust. So the conidia, which are the spores, grow on a hypha that has septa, and it has many shoots in the different segments. It looks like a balloon. Okay, you can see in other photos. Can you please show it?
Yeah, are these pretty big?
Yes, they are. And this is a 100x photo just to show you how it is. That's the crust. It's not too thick, and it's all like space. It's cottony and separated. Yeah. And I have a better detail. Okay, so you can see all the little tips.
Maricel, actually, that reminds me very much of a slime mold that Virginia showed, I think 2 weeks ago, also with this pink color, and that the spores were what I thought were attached at different locations like you're showing here, but she's saying they were not attached. So that's curious.
Oh no, and this red comes from a chemical, Congo.
Ah, okay.
Yeah, I do it so I could see the shapes better. No, this is different. The spores from a slime mold are round. This one looks like kind of an egg or a lemon. Yeah, it's a little different.
Gotcha, thank you.
Virginia says that the capillition on the slime molds are just like structures to hold the spores there. These ones, the spores are born from these little tips. This is the actual, oh gosh, I can't explain. Fruiting body.
Oh, I'm understanding.
Fruiting
Thank you.
body. Yeah, that's the actual fruiting body. But because it's across, it's like not too much to see. Yeah. All right.
Pretty cool. These have like the same type of shape as some of those other Butyria basidium. Like the ones that form the chains. You know what I'm talking about?
Yeah, the anamorphs of other crusts, the Botriobasidium. Yeah. Sometimes they grow in chains. In this case, they didn't. They were coming from that central hypha.
Right, from these.
Yeah. And it's very tiny, like maybe 1 inch or 2 inches, if much. I guess they like the last stage of the last type of fungi degrading the rotten wood, I guess. Yeah. All right. Thank you.
Were these hardwoods?
Yes. Yeah. I was not close to any conifers at all.
Cool. I think that was all of them, right?
All right.
Yep. All right, cool. Thanks, Marisol.
Thank you for listening.
Yeah.
Yeah, I'm here. Okay, I got a couple mushrooms from the Pine Barrens and a couple mushrooms. The other one is a couple mushrooms from Kittatinny. This one I found today. It's a I'm pretty sure it's the used to be a hygrophorus, a hygrocybe, and now it's a Gliophorus irrigatus. It's usually a little darker, but it was so slimy and sticky and gooey that I'm sure that's what it is. The next one shows the very white gills. Very pretty gills. See? And okay. And then the next one.
So there's the name of that one.
Right. And then the next one is, I'm pretty sure is the Amanita elongata. It has yellow on the base there and a tiny little bit of a ring there, veil. But Igor will probably correct me if it's not. So.
Need to measure spore dimensions. The term elongata refers to the spores being more elongate than Flavoconia,
Well, it's not Flavoconia.
which it resembles quite closely.
Except it has a white. This one has a white stem. I've never seen Flavoconia with a white stem.
Okay, not Flavoconia.
Flavoconias always have yellow stem and yellow veil.
Yeah, okay, I see. Okay, I see what you're saying. Yeah, that it's pretty old though.
Yeah, well, it was
old.
fresh, but it was kind of hot today, and it kind of dried out.
Yeah, elongata. That that could be it.
Okay.
So there's that name.
Okay, this anosophy, now I thought it was also, Marcel said it's an Endosperma calamistrata. Is that right? Is that how you say it?
This isn't Calamistrata. Calamistrata is really dark and it's really scaly and has blue-green stain on the base.
That's
Find
what
it
I
in
was wondering
mud
about.
or on a creek bank. It's not
Okay, yeah.
Calamistrata. This looks a little bit more like unicolor, but I don't think it's that either.
Okay, the spores were 5 by 8, 9, and 10.
What about the shape?
And they were round,
Angular?
Ellipsoid.
ellipsoid,
Oh.
except that one side was flat. It kind of
Yeah.
like a little bit like a crescent moon, sort of almost.
Yeah, they usually, a lot of times they'll say bean shaped
Yeah.
for those. But that's not Calamistrata though.
Okay, yeah,
Nah,
I didn't think so.
nah.
But it was very scaly. The cap is really beautiful. Very beautiful.
Yeah, the cap is breaking up into very small scales.
Yep.
You know, pretty much like unicolor. But you know, it's really funny. The physical morphology, like for instance, the way the cap looks on this, does not seem to correlate very well with placement in these new genera. You have to look at spores. Spores are one thing. Look at gill material and find cystidia. Presence of pleurocystidia
No.
is a really
no,
important feature.
No presence. No.
Yeah, well, there you go. No pleurocystidia. All right, that rules out genus Inocybe. I'm pretty sure.
Oh, okay.
So it puts it in one of those other new genera.
Oh, okay.
And yeah, because there's a few of, there's more than a few really. No pleurocystidia, lots of chylocystidia that form like these like barricade type things. I forget the word again.
But I didn't see any crystals either.
Parisate.
No crystals on the chylocystidia.
Oh.
That's only on the pleurocystidia.
Okay.
Yep.
Oh, Marcel collected this.
No, I didn't.
You didn't? Oh,
No,
you
I found it on the table and I was going to take it. I was going to do the micro when I didn't have it. Yes, so I got a lot of photos, but you did the micro. Yep.
Yeah, right.
Somebody, I have no idea who. Yep.
Anybody got a guess on this? I don't know what it is.
I think unicolor might be a Malocybe now. Don't quote me on that though. That might be wrong.
It is.
You know what?
Yeah, that's right.
That's right, Luke. Okay.
What? Look.
Check out Malocybe then. See, maybe you can get a match there.
No.
Luke was right.
Mm.
Hello?
Yeah.
What did you say, Luke?
Oh, I just said, he said that he thinks it's unicolors and Malocybe, and Igor and I both said yes at the same time.
Yeah, so that's at least something to check.
Okay.
But there's so much morphological and microscopic crossover among these new genera. It's a little bit confusing. I haven't straightened it out yet in my own mind. But check out Malocybe. Start with Malocybe unicolor and move on from there. See what you can
Another
find.
genus, you mean?
Yes.
Yeah,
Yes.
Inocybe is now like 3 or 4 different North American genera.
Okay, I
This
get
is what I was talking about before, Maricel.
Yeah.
And
Yes, I get it now. Okay.
I divided up Inocybe into about 5 different subgenera, but he's
Got it.
individual genera.
Mhm.
But they
Got
all
it.
look the same.
Dave, a quick question. You don't think that mushroom that you were talking about is a cystoderm of some kind?
No, we thought about it, but it's not.
This thing? Nah, that's not a cystoderm.
Because we found something very similar looking at Horseshoe Bend, which I took home and dried, and we'll post
Yeah,
them.
it looks a little bit like a cystoderma, but you wouldn't have the brown ornamentation on the stipe. It would be more cream colored, and you can certainly tell from spore print color. Schistoderma is going to have white spores.
Okay.
Schistoderma tends to be a little bit later occurring too. I never even find any until late August.
So what is this? I thought maybe it was some sort of Cleidocybe, but Cleidosby, but I don't know what the heck it is. Any ideas?
None better than that one, really, from me.
Yeah. It was real small, but it had this neat stuff at the bottom.
Yeah, you would think that that neat stuff on the bottom would be a key, you know, and you might, if you check around enough, you might be able to find a name, you know, that that comes, you know, from that particular trait, because that's really pretty striking, really, you know, how bottom of the stalk is. So, I mean, the only thing I would say is Leucopaxillus is known to have this mycelium that connects the fruit bodies underground, but that doesn't really look like any kind of Leucopaxillus. It's too small.
Yeah. Huh. Okay. Well, thank you.
All right, cool. Thanks, Nina. Okay, so I'm next here. So these are 5 things that I saw over the weekend. Okay, so I put this up because I specifically wanted to ask about the Gyroporus. This is what I would always have called Castaneus before, but I knew that paper came out last year that named, what is it, Borealis? And I was just unclear whether, do we have Castaneus here in the United States?
No, we don't.
Okay, so are there any other Gyroporus around here that look like this, or is this Borealis?
Smithii.
Smithii. See, I thought Smithii was more of a Midwestern thing.
Well, either way, you have to sequence these. I don't think that the spore measurements or the morphologies are really going to be conclusive on these Gyroporus species. They all look the same pretty much, and they're variable. And I mean,
okay, so we could probably go.
We'll have to link the DNA with the morphology, and you have to see, you know, we have to sequence a few collections and then see if there are any trends.
Okay, so it's pretty kind of unresolved what we really have around here then.
Yeah, I've been calling these Castaneus group, but I agree with Igor. Smithii seems to be the most likely sort of candidate based on the pale sort of light brown cap color and the stalk, you know, having also a pale color. I find this darker one around here, and I haven't found it in the last few years, and I've been hoping to get some to be able to send to somebody who might want to do a little more work with it. But
if you look at the paper by Navid Davudian, the last student of Roy Hollings, whose area actually is Gyroporus, and I think he's still continuing with it. If you look at that paper, I think he published 2 in the last 2-3 years. Either Smithii or Borealis is closer to the European Castaneus. So in essence, you could be half right about calling it Castaneus group. I have to look up which one of those is in the Castaneus relative, but I'm pretty sure it's Smithii. But
okay, I see. So that's a half right kind of thing, because it might be the one that looks like the other one, but it's not related to
exactly
European Castaneus. Well, you know, as per the usual thing with this stuff. Right.
No, we need to sequence additional collections because it is possible we have more than those 2 species.
Yeah, I've got a nice collection that I'm gonna use as for some fundus that I just collected. But I'm hoping to find this darker capped thing, and I found one earlier, but it was bug infested. It wasn't really, you know,
the only problem is with sequencing is that the Davudian's paper, I think, uses loci other than ITS to do the phylogenesis.
Oh, okay. So you know what?
We have to give it to
then. Yeah. I'm glad you're telling me this, Igor, because maybe I'll put those aside and just send them right to him.
Yeah, I mean, getting ITS would be useful too, because that's a more common genus that people sequence. And
I see.
Gene or locus, I should say, that people sequence anyway. But
Well,
RPB is more exotic.
I
And
can always take a little sample for Fundus, send it in, and leave the rest and send to him.
exactly. I mean,
Yeah,
you put a small sample in a test tube and send it off to Fundus, and they do.
that's right. That's what I think I'm going to do. I've got a nice sample of, looks just like this one. You know, oak woods, mixed woods. I just posted a few hours ago. Really?
Okay. Yeah, that's what these were.
How big were these? They look pretty small. What are
Yeah,
they?
they're pretty small.
Yeah, these were
Cap diameter rarely exceeds 2 inches.
Yeah, they were pretty small, 2 to 3 inches tall, hollow stem.
These are very good pictures.
Thank you.
Yeah, those are nice pictures. I agree. Yeah, useful picture.
Okay, so I did not collect them, but now that I know that we should be collecting them, I'll be a little more diligent with them. These guys, I put these pictures up because these were so pretty. So Boletus separans, these were really in nice shape. A little blurry on the top there, but.
Yeah,
now, there's no doubt about the reticulations on those
That's
types.
beautiful.
It is possible that we have in the Northeast more than one species. I know there's more in the USA. There's a separans group. We have 3 species in there, but morphological variability, reticulation, dark cap, it suggests it could be something other than our true Northeastern separans, but it is in the separans group. So this is the best idea.
Igor, I was reading that if you put KOH on the cap, it's supposed to turn green. Is that true?
Not KOH, ammonia.
I mean ammonia.
Yeah, ammonia. They will all do that.
They all do the same thing. Yeah.
But I had something the other day that sort of looked like this, and I put ammonia on and it didn't change. So that would mean there's something else, right?
It could be slow, and you should put ammonia on the stipe more than the cap. And if the cap is not lavender, if it's brown, it's not going to stain green with ammonia. That's what I think.
Yeah, right. I agree, Igor, because I've had some that were faded, and I didn't get a reaction.
Or it's very slow and faint. Look, you can call these whatever you want. We can call them separans and call it a day, and it's probably true. But I just want you to be aware, that we possibly have more than one entity even here in the Northeast. The other one, which I called pseudo separans or the imposter, it's more of a southern thing. You find it like in North Carolina and Texas, and it could be here in New Jersey. But we'll need to obviously do more sequencing and comparing morphology and things like that. But as far as I can say, these are separans probably.
All right. Well, we're not sequencing these because I ate them.
Oh, you ate them? Oh,
sorry.
I just picked some with
look how nice they are.
smooth stipes that I'm going to submit. At least a small sample.
Yeah, I found separans recently that had absolutely no reticulation, nothing.
Yeah.
And it was more pale too.
Okay, what do I have next? Oh, I put these up. I haven't seen these yet here on Taxonomy Tuesday, the Galliella rufa.
Oh, yeah.
They're pretty cool. They're super abundant right now. So didn't
you had some wet?
Yeah. It's been raining.
Didn't we see a bunch?
Love to find those.
We saw tons of these at
Horseshoe Bend. Okay.
So this is the fertile surface. This is the side that we see when you flip them over. This is kind of what they look like on the underside. So these pictures, I don't think really do them justice. But didn't you guys say, the things that you were showing all spring, Susan, the ASCOs?
Sarcosomas, yeah.
Sarcosomas. Yeah. Didn't we say that these are pretty close related?
Yes.
Yeah. Actually, when I first
like them at least.
When I first saw them 8 or 9 years ago, I said, "That's a Galliella rufa, except that it's backwards for color." My guys have the disc, inner disc is black whereas the outer rubbery walls, so to speak, are a dark brown, whereas here yours are light, light brown, whereas here it's light in the center and almost black underneath.
Right. So we know, yeah, they definitely look alike. Do we know, are they related?
They are related, but they are still separated. They could be in the same clade of
yeah
genera. I mean that probably could be in the same family or subfamily, but they're not like sister genera. They do have some separation between them, but pretty much they are close as you can get.
Cool.
Yeah.
So it's not some weird convergent evolution.
Right.
I sent
So did you cut through any of them, Luke?
No.
And it shows the relationship.
No, I didn't cut into them.
I can send a paper to you too, Luke. I have to find it.
Yeah, I wouldn't mind seeing that.
Yeah, you should be able to find a few more of those. There should be a lot of those with all this rain and this time of year.
Yeah, usually a lot of them come out at once and they're always attached to some kind of wood.
Yeah, cut through one of them.
We found a bunch
inside.
Kittatinny. Yeah, they're very gelatinous. There were several on a stick. Maybe the biggest one was 1.5 inches. Well, it was pretty big.
We're going to find a lot in Middle Woods next Saturday.
Okay, this one I showed of Radiolomyces last week, the Copelandii. So this week, I actually found Radiolomyces copelandii and this one, Palmanokensis, on the same log, like literally on either side of the log. I've seen that a couple of times, but I'm pretty sure we're looking at two different species here. These guys, the Palmanokensis, are much more rounded. They come from like these orbicular kind of structures, and they tend to branch, and they have this orangish color to them. So if you guys remember the ones I was showing last week, the Radiolomyces, they were like straight up and down, like icicles. See these, how they kind of start from like these like globose structures and come out.
They look almost like antler chandeliers.
Yeah, yeah. Yeah, and these, they're kind of divergent. If you look at like this one right here, it branches several times. So this is a new name. This was just described like a year or two ago, based on Long Island. This is a Native American name for Long Island. Oh man, what is his name? Kara Nakasone was involved.
Okay.
And there's a gentleman, I'm trying to remember what his name is. He lives on Long Island. He often works with Herbert Baker.
Joel Harmon?
Yep, him. Joel Harmon.
Yeah, okay.
Yeah, yep. I think it was those two, Nakasone and Harmon. So anyway, this was cool. I didn't include the photograph. I tried to take a picture to show two different types of Radiolomyces on the same log, but it just wasn't, the picture didn't come out neat enough, or, well enough to demonstrate it, but they were literally growing within a couple of feet of each other on the same log. And the last one I have is a really common polypore, but this one was in better shape than I usually see it. Usually you find the old dried ones. For Fuscoporia gilva. This one wasn't super fresh, but it was still fresh enough that you can still see the distinctive yellow bands on it.
Yeah, the first time I saw this one, it was fresh. I didn't know what it was. I'm used to seeing it when it's a lot drier, thinner, and doesn't look the same at all. Used to be Phelinus gilvus.
Yeah, when they're absolutely fresh, I mean, they are stunning. Like this, probably probably a week beforehand was absolutely stunning. They're starting to dry out, but they always have this mustard yellow band on them when they're fresher. When they dry out, that mustard yellow goes away. And the pores on them, you can barely see the pores on there. The pores are really tiny, like 6 to 7 per millimeter. Sometimes they're so small you can barely see them, or you can't see them without a glass.
You
I
can
think I
use,
might have an old mushroom observer post on this species, although I might be under Philinus.
yeah, it would be years ago.
Yeah,
Yeah,
it's only been a couple years.
Which was a really, which was that a nymph foray, actually, I believe. Or a Nama, maybe, I forget. Yeah, they look really cool when they're fresh. I know that. I don't
Yeah.
find it very often.
Do you ever dye with these, Susan? I've read that they
Yes.
do dye.
Yes, you have to use kind of a lot, and it's better when it's fresher rather than dried. But you can use it to dye wool. Alum would be the deep gold.
All right, cool. All right, well, those are my 5, and then Dave is next. I did want to check in. Is there anybody else that has mushrooms that they want to share?
Lila here. I'll share a few after Dave.
And is there anybody else?
I can, but I don't have to.
Okay, well, I think we have enough time. 3 people. I mean, we have.
What time is it now, anyway?
It's 8:30, so each one of you takes 10
I'll
minutes.
try to go. I'll go fast.
If each person takes 10 minutes,
I'll try to go really fast. I got a lot of things here, but I'll go. Let's go fast. Some of them won't take much time.
Speed taxonomy. Oh, good. I was hoping you'd have this one up.
Yeah, what the heck is this?
I don't know. I was hoping you'd have a name.
Some kind of Craterellus. I mean, there's this what is it, Phoetidus or Odoratus? I get them mixed up because they both mean smells. But it didn't smell. You know what? There's a few other Craterellus species out there, and it really, you know, I'm sort of retired now. I'm not going to really go back to work anymore. My retirement officially is October 22nd, but I'm busier than I ever was now, you know, basically because of all these mushrooms. It's pretty interesting though, just a pair of them growing right next to one another. They're pretty clearly the same species. One is mature or maybe post-mature. One is slightly immature. Got spores from both of them. They look the same. No noticeable odor. Pretty interesting species. I'm not expecting any. If anybody has an idea, hey, put it forth. But I'm not really expecting a whole lot here. I'm maybe gonna have to do a little probing on this one. Who knows? I've got it preserved. Now, fundists might not want it because there are certain genera that they don't really want to deal with. And that might be one of them. But if not, I'll send it to them. Now, this is a common mushroom, but not in my area. I never found this up here in Northeast PA, ever, until this year. It's the green spored parasol mushroom, which is toxic.
Chlorophyll molybdates.
Yeah, molybdites, I guess. Yeah, I've never found it. I found it in Binghamton once, oddly enough, which is north of here. And it probably was a really hot summer because when I lived in Binghamton, there were a few really hot summers. The gills are free, but that's not a very particularly good rendition of this because it looks like a slug who was risking admission to the ER was eating this thing. And you see, it's got this ring that's clearly looks like I could detach it and slide it. But the big thing is the next slide. I believe it's the next slide. There it is, green spore print. Yep, not a white spore print. So that makes this molybdites. Excuse me?
Is that like a cutting board sort of that you did the spore print on? What is it?
Well, a friend of mine made this for me a long time ago. He's a sign maker. He's got access to all kind of really cool materials to make signs. So he took this piece of pure white polypropylene plastic or whatever, and took this really high grade, really thin electrical tape, which is black, and made strips. And it's pretty cool. You know what? I will take a picture of the underside of that thing for next Taxonomy Tuesday, just because it's actually kind of funny. And actually, he made another one too before he made this one. But next week you'll see. Yeah, the green spore lepiota. Let's move on. That's not a particularly interesting mushroom, but it's interesting that it's growing around here because I've never found it around here.
I think it's interesting. It's one. It used to be one of the biggest things that was a gastrointestinal poison in New Jersey.
Yeah, every year it leads the list on the NAMO. It's on the top of the NAMO list. Yep. All right, here you go, Luke. So is this some sort of notice, or is this mysterious Bugliosporus thing, or whatever it is?
That's what I think it is. Bugliosporus.
Yeah, I wondered.
We got to see the underside.
I wondered. It was on oak, no bark.
Yeah.
Staining.
Yeah, that's Bugliosporus.
Oh, good. Okay, I got some spore pictures. I didn't preserve any, unfortunately. I let it sit out on that spore board too long and it started getting moldy and soft, so I just threw it out. But
it's named after.
But that's a little further north.
Yeah.
A little further north and west, So, yeah, I thought that might be what it was. If you blow it up there, you can see the spores. Kind of like marasmiella spores or something. Yeah, they are certainly from this thing.
See that color, that staining?
Mm-hmm.
That the name Blugoso has something to do with ox tongue.
Oh, okay.
It's named after that flavor.
Okay.
Did you taste it?
No.
It was one of the most horrible tasting mushrooms I've ever tasted.
Well, okay.
Like bitter,
I'm glad.
Bitter to like.
Yeah. Okay. Good. Okay. Maybe I'm
looks
glad
a little
I didn't.
like fistulina.
Yes.
Yeah.
I thought there was this other kind of fistulina. I thought that might be a thing. There's also these innonotus and these pseudo innonotus things that seem reasonable for this, but in fact, I put it on Mushroom Observer and I said polyporales, which basically means like duh, it's a polypore, and I also counter proposed innonotus because that was the closest thing but I really thought this might have been this thing that Luke's been talking about. And so good confirmation. All right.
I'll roll it out right now.
You go ahead, Luke. Propose it on there if you want.
I will, but not right now. I won't waste.
All right, I'll let you do it because you're the one who knows what it is.
Luke.
Yes.
You submitted Bugliosporus corsinus for sequencing to Fundus, and I have a sequence for it, which I processed today, and that looks pretty good, and I think that's what it is. So,
awesome.
Your ID was correct.
Nice.
Excellent. All right, more confirmation.
Okay, everyone see the name here? Still there, Dave?
He's muted.
Dave, you're muted.
Sorry, talking to myself here. So there's this peck species, Boletus vermiculosus, named for the tendency of the mushroom to be loaded with like maggots, vermin. And so then this other species came along later on, vermiculosoides, which supposedly looks like vermiculosis. And so I kind of wonder, are there really two different species here? But anyway, this one, beautiful specimen. My wife just happened to be like stopped at some rest area to eat a Subway sandwich, and this thing was there, and so she picked it and brought it home. And it's not like it's an uncommon species or anything, but I'll tell you what is uncommon. What is uncommon is to find one of these in such immaculate condition, because when you see the next, look at that. No, vermin. This thing has not been penetrated by any kind of bugs or anything. So this goes to fundus. This one will
Oh,
go
that's
to fundus.
awesome.
Yeah, it's pretty cool specimen.
Beautiful.
Yeah. So we're trying. There's a lot of interest really in this vermiculosis, vermiculosoides, and then there's this other one. What, Igor? You might be able to correct me here. Sub something or other.
Subgraviolens.
Yeah. The one that the Bissets said, and I quote roughly, when dried, smells like boiled urine.
Well, that's what Smith
Not
said.
sure how anybody actually came up with that, but it's this probably not that, but it looks like this pretty much. I'm guessing that the species really is that they're all vermiculosoides, and who knows what the heck Peck found.
So is this with oak?
Yeah, pretty much. Yeah, there was oak in this area. There was oak and some sort of pine that she didn't know what it was, and if she didn't know what it was, I would have to work at knowing what it was because she's better at trees than I am. Some kind of like 2-needle pines that are planted there, but it's oak. It was probably with oak. I find this in a lot of places. Usually with oak. You know, it's funny. Actually, now that she questioned, I usually find this in areas with oak, but there are like conifers present, like planted pines and things like that present. Now it might just mean that that it's good to have like an open area that gets a lot of sun, and the oak is actually the main thing going on. But I rarely see a specimen of presumably the same species. Maybe there really are two different species. That's so free of insect damage.
I used to see that a lot in the place where you and I both know about.
Yeah. I've made several collections over the years. I can actually say that I've sequenced material with brown cap and the yellow cap, and I think I sequenced 3 or 4 collections, and they all came out to be the same thing.
Oh, interesting.
Yeah, whether this is
Mushroom.
Smith's vermiculosoides or Peck's vermiculosis, I don't know. And maybe we'll never know. But Peck's descriptions are very scanty and wanting. I don't like the way he describes species. He did a lot of misjustice for us because he confused everything. He described species with 2 or 3 sentences pretty much. That was his descriptions. And then his types are so old you cannot sequence them anymore. They're gone.
Yeah, I would believe that. Yeah.
So,
well, back in defense of Peck, maybe back in 1875, it didn't seem like there was that much of it, necessary in terms of all sorts of information. I don't know.
Yeah, but he should have thought about future generations, that there will be more technology and people should be able to match what he found with what they're finding.
Yeah, well, that's being some sort of like a myco-visionary.
And if he's writing a flora of New York, that should be included with more information.
I agree, though. I will agree with you, Igor. His descriptions are scant. There really should be more details.
Look at Singer's descriptions, and even Murray's descriptions are better than his, even though they were.
Yeah, well, they've come another 50 and another 50 on top of that, plus years ahead. It's I mean, back in 1870, it was like revolutionary, that there was something in, that maybe Boletus was not all one thing. I mean, it's this historical context kind of thing, really.
All right, you better pick one more, Dave, and then we got to move on.
Yeah, I'm sorry, I'm taking too much.
Any of these?
Just go to the next one, whatever it is. Oh, just this one I included just because it is so out there. look at this basal vulva on this thing. At first I thought like somebody dropped a piece of cardboard or something on the ground and it just kind of wrapped around this thing, but that is actually the basal vulva of this mushroom seen on the bottom part of the picture. I don't, my guess is this is probably Amanita Westoniae, but you know, as Igor will confirm, there's really a need to have these Amidella, section Amidella things sequence get this stuff straightened out. But certainly this one is an anomaly, and it's worth submitting for sequencing. So I preserved it.
I just got 4 sequences of 4 species of Amidala sequenced by Fundus. I got LSU from Linus, and Fundus gave me ITS. None of those species are in Rod's database. None of them.
Well, I've got another Amidala I'm going to send in as well, a little one, a very small one.
Bets are off. We don't know what these things are. It is possible it is Westoniae because of the tubular, long vulva.
The long limb. Yeah.
Yeah, it's more like a test tube than a cup. But
yeah, no, good description. More like a test tube than a cup and one long limb. Yep. So my guess is it's going to come back as Westoniae, but who the heck knows? And I'm putting it in.
Yeah, go ahead.
Really good specimen.
It's going to be a good data point.
Great pictures.
Yes, that's what I'm saying. Right. Yep.
Cool. Well, thanks, Dave. All right. Violet, you want to jump right in there?
Sure. All right. For Bianca's viewing pleasure, this is the Thalius schweinetzia. And Susan will like this, although we've got 3 boxes already.
Well, you're not going to see a nicer one than that.
Yeah.
I just saw a paper, and I think it was through Mushroom Observer because somebody brought it to my attention, or I just saw it somehow, that we don't have Phayalo schweinetzia in this country. We have other things.
Ooh.
It's a European species. So what we have here is our own Schweinetzii, quote unquote.
Interesting that David Louis de Schweinitz is an American mycologist buried in Moravian Cemetery in Bethlehem, Pennsylvania.
Right. Von Schweinitz, he was German.
Right.
Do you have that paper, Igor?
I may have a reference. Maybe it's not free online, but I'll try to find it.
Wow. Makes great colors, yellows and golds with alum, and a nice kind of an interesting green and almost a brown with iron. And I haven't done it in years, but with copper, it's supposed to be chocolate brown. It's good with every mordant as a dyer, and you don't need a lot. I always use it from dried.
Okay, hence the common name dyer's polypore.
Correct.
Yeah, these photos are field guide quality.
Yeah, it's really good.
Thank you. Early morning, hazy, humid day seemed to work.
So did you pick it and keep it? Or?
No, I think it's still growing, so.
Well, but the thing is you see how it's gotten dark in the center?
Okay.
It stops and starts growing. I would still pick it if you're going to use it for dyeing, because for the most part, you're going to want to use about the outer 3 inches. I would tend to not use the very dark in the center because it will be a muddier color. You want to use the very yellowy growing edge and only about 2 to 3 inches back.
All right, so should I pick it and trim it and then dry it?
Yeah. Sometimes I just dry them whole because you can do that. But if it's thick and wavy as it looks, break up the fronds and you can even cut it up and dry it on a food dehydrator.
Okay. All right. I'll go back tomorrow morning and it's probably still there. I'll get it. I'll save it. I haven't dyed anything, but I'll save it. Somebody else can use it.
Yeah, there's probably several in your group that would like it. A shame to cut it up, but still.
Yeah, yeah. Yep. Very, very pretty on a kind of gray, hazy day. It was, oh, there's something to look at. Okay, so that was that one. Here's one that I didn't seem to get a picture of it in situ. Found it on the 4A Saturday. And I don't know what this is. I don't know if anybody ever did get a name on it. Ursula was there. She says it's a Cortinarius. And I was just fascinated by this, and I call it cross veining that's going on in here in between the gills.
That's really something to see.
Yeah,
You
yeah.
have a picture of the top?
I do. There's the stipe.
Yeah, I would tend to agree, Cortinarius. I don't think I would bother using it for dyeing, but then she's more apt to use a lot of things I wouldn't use.
So
The widely spaced gills suggest Cortinarius section HINNULEI, I think, section. But these subspecies and sections of Cortinarius are now viewed with suspicion because they were carved out via morphological or micro traits, and so DNA is not necessarily going to support these distinctions. But the gills look kind of widely spaced, and the cap looks kind of grainy. And section Hinnulei, I guess is how you say it. So one of the main species in section Hinnulei is Cortinarius distans. I don't think this is Cortinarius distans.
No, it's not.
Yeah, I looked at that one somewhere, and it didn't seem to match.
But it has some of the same characters. Now, if you can get a spore print, look, if you want to send spore, if you want to mail spores to me, I'll look at them under my microscope. And the section Hinnulei seem to have a somewhat distinctive spore morphology, fairly warty, varicose, and kind of small, and not a real big length to width ratio. But anyway, if you want to send me an email, if you want to, I'll look at spores. For anybody who wants to send me a spore sample, I'll look at them. My microscopy skills are not great, okay? But I do know how to look at spores, and I can get decent measurements. Go ahead. I'm sorry.
It's gone. It's rotted. It's stunk through.
Okay, so it's gone.
But one thing we did, Cortinarius, we did find was Marylandensis.
Oh, it's gone.
Very, very beautiful.
Did you really find Marylandensis?
At Ursula, yes.
Nothing's going to bring it back. Are you sure it's not?
No, it's not sanguineous.
Two other of those red ones. Let's see it.
It was large. It was fairly large. It was orangey, reddish. And we looked it up, and yes, Ursula grabbed it to take it.
Yeah, I would think she would. But did anybody take a picture?
No, I don't think so.
That's too bad, because I'd like to see a good picture. That's kind of a phantom mushroom.
Do you know if it was one of mine? Because I took a picture of my whole table.
I'll say it's interesting that you guys found this weekend, because I saw a photograph from Philadelphia about a week ago of something we were calling the same, Marylandensis.
I'd like to see a picture of that.
Yeah, it's totally red.
Maybe Bianca has it. We'll have to see.
Red top, red gills.
Yeah, but it's not like dark red like the
pinker.
It was more of a it was lighter color.
Yeah, it's pretty vividly red.
Right.
like scarlet soda red.
Wow.
All right, here's the last one and then turn it over. Some sort of Lactarius, very short site. The enrolled margin, zonate, white latex, and I don't know, I tasted it, it was very unpleasant. I would say peppery, but maybe it's acrid. Was not a pleasant taste at all. It seemed to have, Marisol was interested in these pits. Both in the cap, you can see it kind of pitted, and this was almost completely buried by leaves in a pathway. And I just bent over to see what it was, and there it was a mushroom coming, forcing its way up through the leaves.
Can we see the top again?
Sure. So I think I might be, what is it, Samicola?
Nah, Samicola is zonate.
You don't think this was zonate? It looks zonate to me on the edges.
Nah, zonate means there are concentric rings of different color on the cap.
So this dark and light is not.
Dark, yeah,
Dave.
dark and light, dark
Yep.
and light, that sort of thing. Yep.
Look at
Yeah, I
the,
don't know. I have no proposal on this one, but I don't think it's Samicola. I think Samicola might have. Okay, you know what? I think I might have the Milky book right here. I think Samicola might have closer gills too. Let me check that.
Doesn't Samicola also have a more fruity smell, a little bit of a smell, which is not unpleasant?
That's a funny one.
Drops on the stipe. I remember about Lactarius that has that feature. It's like these
Yeah, the
spots.
potholes on the stipe.
Yeah. There's one
Yeah,
species of
that's a
Lactarius
feature.
that has that, if I get its name.
No, there's like several species that have that.
Okay.
Yeah, but what was? I'm looking up Samicola. I got the Lactarius book here, the set thing. I think Samicola, the gills are close. Gills close. Yeah. Okay. Other kind of Samicola. Gills close. Yeah, there's like two varieties of Samicola they have here. The gills are, these gills are not close. They're subdistant to distant. So I don't think this is any kind of Samicola. I don't know what this is though.
Okay.
I really hate to say no, it isn't, but not have an alternate proposal. But that's as good as I can do right here.
Now that was as close as I could get, but I wasn't very confident that I had it right.
Yeah, you know, put it on Mushroom Observer if you can.
I'm gonna have to learn to use that tool, I guess.
Yeah, well, it's a real big bother. Don't worry about it. And I'm not going to be able to figure this out probably anyway, honestly.
All right, so
yeah, there's too many online forums. It's like everything gets more and more and more spread out. Mushroom Observer, iNaturalist, Facebook this, Facebook that, Facebook the other thing. It's not good. It's not good. That this whole thing should be more centrally situated.
All right, so guys, we're running out of time, and Bianca still wanted to share hers. Then Susan's
In the
asking
chat,
for.
there was something about, I think Lauren was going to show a particular mushroom that you guys are talking about. So
What do you guys want to see that first?
I would like to see that. I don't have to.
We're talking about the Cortinarius, right? The Maryland densis.
Yeah, it was Lauren. Lauren took some pictures. She got it.
Oh, she said she'll show it next week.
Okay.
So I'll say right now, because it's 9 o'clock, and I just want to, if anybody's ready to go, I want to thank everyone for joining us and all that fun stuff. But we don't have to leave quite yet. But I just want to wrap up in case anybody wanted to get out of here. So thank you everyone for joining us. We'll see everyone next week. But go ahead, Bianca, you're in overtime now.
Yay, overtime. All right, so I have a bunch, but I don't know, just like what was said earlier. Hold on one second, let me. All right, so I had a bunch of mushrooms that I had found, but again, like it was said earlier, I don't know which ones are particularly interesting versus not interesting. So how about I just kind of scroll through them and you tell me stop and we'll talk about it. Does that sound good?
They're all interesting.
Yeah, exactly. Well, to me, they are. All right, let me get to the beginning of this list here. I already did that one. All right, I will show my screen. This one. All right, so this one I had already shown. Let me just make sure. Oops. All right, this one I had shown. We talked about it. This is the 21,717. Okay, so this is a bolete. It looked on the older side. If I remember correctly, it had a little bit of a foul smell, not too much, and it was a little bit bitter on the taste. All right, so we'll move on. So this is all the same mushroom. All right, moving on. Then there's this one, which was also on the older side, but it was very unique on the gills. It looked very wormy. I don't know. What is it?
It's hypomicized. It's a mold.
This is mold. Ah. Wow,
And
no
dis-
wonder.
And distorted by the mold.
Go back a couple pictures.
It's Xanthoconium athene. That's what it is.
Yeah, I was gonna say it's probably a xanthoconium.
For this first one?
Yes.
Yeah, back here where it's you can actually see what it is before the hypomyces gets hold of it.
Okay, so this is the second mushroom. All right, that has this mold. What was that? Was
Yeah, once the mold sets in, it becomes something really different. Hypomyces, I think the species is Trisospermus.
Chrysospermus, yes.
Yeah.
Yeah, it's the same mushroom. It's Xanthoconium athene.
Oh, so that was the before and this is the after.
That's what I was going to suggest the same.
Yeah. It frequently gets hypomicized or gets moldy like that. It gets moldy.
This species gets attacked really easily.
It distorts the shape and it smells pretty bad. Very
Oh,
fishy.
yes, it did.
Very fishy, very stinky.
Yes, absolutely.
Especially in damp, rainy weather.
Mm-hmm. Does somebody mind putting the name in while I continue?
I'll type it in.
Thank you, Igor. All right, so this is the Arusula, very gorgeous, nice color. Hold on, the cap you can see is like a ruby color. And it has a little bit of pink also on the stipe. Oh, that's a different one. So those are the two. Oh, and there's a little one also you can see on the left here of the same. Okay. So I will move on. Then there's this guy. This is, I knew the name. We went through it before, but it has that sac that it's in. And it was this kind of grayish color with the, I'm not remembering the terms, I'm sorry.
Striations on the cap margin.
Thank you. Let me see. There's
Amanita rooseveltensis.
Yeah, it could be rooseveltensis. Amanita section vaginate would be a very general distinction. Amanita stirps rooseveltensis would be a less general distinction, but I agree with Igor. It's very likely the species Amanita rooseveltensis. but we're
Okay.
still trying to completely understand this species, really.
Right.
Oh, okay. Well, you mean the amanitas in general, or what do you mean?
No, just
Guys.
well, amanitas
Very
in
well.
general, yeah. I mean, Rod has spent like I don't know how many years of his life devoted to like obsessing over, you know,
What do you want?
Section Vaccinate, yeah.
Join the
in
club about the
particular,
same
just
time
section
I-
vaginate. And it's completely baffling and but yeah, this could be, yeah, this fits the description of Rooseveltensis. But to really be able to say, oh, it's Rooseveltensis, or at least Sturp's Rooseveltensis, I'm you need to look at spore dimensions.
Okay. All right. Let me just, I want to add this into here.
Guys, I gotta bail out. I'll see you next week.
Hi, Susan. Nice
All
seeing
right,
you.
see you, Susan.
Yeah, I'm gonna say
Hey,
goodnight.
Susan, maybe we'll have some mushrooms from the Adirondacks for you in a couple of weeks. So.
I'm gonna say goodnight too.
All right, Dave.
All right, Luke, thanks.
Luke,
Thanks
sorry,
for
Luke, yeah.
doing this.
Good night.
Good night.
Again, I have a lot of pictures, so I don't want to, I'm just going to go through more quickly.
Yeah, just go ahead. I'll stay. I'm staying.
Oh, thank you. But you're not,
Yeah, we
you know,
got
when
a
you're
few
tired.
people staying here.
Hey, Dave and Igor, that vaginate kind of subsect there, is there any way to distinguish between them? Like, what should I be looking for? Because I think I found that exact mushroom. Like,
Could be looking for DNA.
Yeah, pre-DNA, you might want a microscope or use a microscope at 400 power at least to look at the spores and at least see if they're either ellipsoid or globose, more or less. And that's like step one, really. Igor's saying, what Igor's saying is steps, 2 through whatever.
All right, so don't be mad at me, but can you put the name in the chat again, please? I like naming them afterwards so that I have a chance to get through. All right, so this is number 3. Let's see. I pulled it out and the sac remained, so I took a picture of it. So this is another. Oh, did I not take a picture of the cap? Was that it? Oh, this is the cap. This is the cap of it. And there's the remainder. And that's the top, the underside. So that's that. Moving on. This is a bolete for you.
Celopolis, probably mild, maybe not. Oh, lots of worms.
Yes, quite few, but it looked nice from the top.
Yeah, Celopolis you should taste, and you can separate them. Are they bitter? Are they mild tasting? And that's the first distinction you have to make between Celopolis species. But Celopolis species are going to start off with white pore surfaces, and then they're going to turn pink, and they often also bruise brown on the pores.
Yeah.
They're just brown in general to begin with. Have brownish stipes and brown caps, and kind of a boring genus. But there's lots of Celopolis in this country, many more than they describe in the books. So it's actually becoming a very interesting genus, after all.
After all?
Yes, after all.
Okay. All right. So let's see. So here's the next one. This was, I just went back because I saw a similar one to this previous one, to this one. It had the very strong, almost like peeling stipe. So I believe it's the same, but it had a similar cap as the other one. All right. So then this was, I believe, a crust, if I'm not misquoting. And it has these, let me see, this is the underside. Gorgeous
Schizophilum commune.
Schizophilum commune.
You guys are too smart for me.
Yeah, well, look at the one in the upper right center. You can see it has sort of like gill-like things emanating from like a hole down the middle. Right. Those are not true gills, though. There's these like folds that are very sharp but blunt. on the
Upper left.
edge.
Yeah. Zoom in on those two upper ones, Bianca.
Yeah, schizophylum commune.
Okay.
Can you zoom in on those?
Sure.
They're very interesting. I like these. I'm
Yeah,
always
yeah,
pleased
show
to find
close up so we can see this. Yeah.
Let's see if I can get a better. There we go. These might be more
focus.
focused. So it's a little fuzzy kind of cap, but then these.
Fuzzy kind of cap is another feature.
Okay.
Fuzzy cap. Yep. Those are not true gills.
Ah.
They're just like these folds with
Mm-hmm.
blunt edges.
And those are tiny, correct?
They're
Yeah.
tiny to less tiny to not quite tiny.
So
they vary in size.
What's the largest that they grow to, like cap wise?
Yeah, maybe 1.5 inches or so.
Oh wow. Okay, cool.
Yeah, maybe
usually
2 inches would be a really big one.
Yeah, these oblong ones were about almost 2 inches.
Oh wow, those are big ones then. All right.
Yeah, they were comparatively. All right. Did you get this one in chat?
Yes.
A common name, split gill fungus,
Schizophylum
or split gill mushroom, but it's not a true gill.
Gotcha. All right, moving on. This guy, I believe potentially this is the same. Let me see how to describe it. I think it's as similar as the one before with the cap having those
striations.
Striations. Thank you. I'll get it.
Well, the striations could indicate an amanita or a russula.
Right. So this one.
I think those are probably amanitas.
That's what I guess.
Rooseveltensis.
Yeah, it's something like a Rooseveltensis.
Look at that powdery stipe. It's very.
That's not going to be a russula. You're right about that.
Right. Yep. That's why I guessed it. Okay. Let's move on. So here's another gorgeous amanita with a slug. You can see the skirt. And I have a couple more pictures. A little bit eaten. And that's it for this one.
Probably flavoconia.
Yeah, probably flavoconia.
Okay. Let me add that. I'm going to make sure to copy. Okay, moving on. Next. This guy has a trumpet-like hole. The three of them were together. Oh, not this one. So those three. Peach color or like skin tone.
Is John Burkhart still here?
Yes.
Well, that's your department, right?
I can't tell whether is that a russula?
No.
Or
not a russula.
Lactaria? Oh yeah, or it might be a lactarius.
It is Lactarius.
Lactarius?
Yep.
Yeah, it could be.
Volmus or volumus?
What?
No, it's not Volumus.
Nah, it's not Volumus.
What?
Need to see a lot of latex for Volumus.
Look at the habitat.
And the stock should be brown.
Yeah, it's pine oak.
Conifers and hardwoods. It's pine barrens.
Pine oak. Yep.
Yeah, that's not Volumus, not in that habitat. Okay, it's on Planet Pine Barrens. I don't know what this is.
Where did you find this?
This one is one of two places, but it's in the central Jersey area. So it's potentially, this one I believe was in not far from the Manasquan Reservoir.
Okay. Yep.
Pine barrensy.
It's a little outside the Pine Barrens, but there's still a lot of pine oak woods.
Right.
It's not Cantheratus though, right, John?
What's that?
It's not Conferatus. It's not.
Amphiaratus has darker gills.
I don't, was the, what, did the top have a little nipple?
It was actually it was more like a belly button.
No, the Camphoratus will usually have a little nipple.
Okay.
Yeah, even when they're young, they have this belly button.
Looks like it's a fall because it was eaten. It was eaten out by a slug. So this is not a natural cavity.
Yeah, tough to.
The
Galactarius mushrooms are tough to tell as is.
The thing to do with these is to take your knife and run it along the gills, and then see what if it exudes latex, that tells you it's a Lactarius.
Yeah, I usually cut them, but I think I was rushing in this case.
And then observing the color of the latex, whether the color of the latex changes and whether the latex stains. All of those things are indicative of, or sort of.
Gotcha.
I find them really hard to sort out.
Yeah, you might have to wait like 5 or 10 minutes to see what happens too.
All right. Okay.
Do Lactarius generally grow in kind of clusters like that, or is that?
No, that is weird.
Yeah, that's cool.
It's not clear whether they just happen to be close together or whether they're joined at the foot.
I think they were joined.
Okay.
All right. So sure, Lactarius of some sort. I will put that down. Here's the next one.
Well, maybe. I think there's probably other possibilities for that one.
Oh, okay. All right. So here, this is a bolete, small one.
Same as before. Xanthoconium athene.
Xanthoconia. Okay.
Yep. I'm going to type it in.
Athene. Got it. Okay.
Ends in E.
Yes.
Xanthoconium in Latin means yellow cone, which is referring to the color of the spore print.
Oh. Okay, let's see. Moving on.
Which is what? The color.
The spores.
Is yellow?
Not the
yes.
spores.
Xanthoconium has yellowish brown spores.
Yeah. Got you. Thank you.
Okay, so then this specimen was found on the ground. I thought it might have attached to there, but I couldn't figure out how.
Last picture. Is it? This is something else, I think.
This is the same. This is a section of this one.
Well, that picture and this picture, same thing, right? I don't know. Xanthoconium or Tolopolis? What do you think, Igor?
I think this is. Let's see.
Tricky one.
But this is the same as this is the 3 mushroom. This is this one.
Yeah, I think this is Xanthoconium. Yeah.
And this is the section view of that one.
Okay. Yep, you know, they're common in the pine oak woods as well. We find them a lot in the cairns.
All right. Okay.
Yeah. All right, this guy, this is the top, this is the underside, and this is the section. Oh, that's it.
Is that some kind of a Trametes with the moss growing, or the I should say algae, whatever it is?
I believe it was harder than just a Trametes. I believe it was just oh, I forget what they're called. That attaches to the tree directly. I know Trametes do too, but it was stiff. Like
a Ganoderma?
Yeah, like a Ganoderma. See, you can see how hard the wood is, like in the section view. Very dense.
Looks like 3 growth layers there.
Mhm.
Can you zoom in?
3 or more.
The mossy polypore.
That's what I'm going to put down. I prefer it. Next, this is a crust. You can see how it's very small, the sections, but it expands. And then this is with a macro lens, a little bit zoomed in. Should have shown it when, maybe I'll show it next week with Maricel here. She might help.
Yeah, she'll probably know what that is.
Yeah. All right, I'll move on. This guy, it's I believe a Rusula with a nice color, peach color top, yellow. I tried to do the distinctions, but it was so difficult to do. Some of them just say yellow brown, some say pinkish and reddish, and it was, I'll get better at it.
I know what you're thinking.
You do? What's that?
I know what Dave is thinking. Dave is thinking this is Modesta, right, Dave?
I'm back. Oh, wait. Okay, let me look. Nah, too pale for Modesta. And Modesta doesn't have an umbo.
It's just
No,
an
I don't think this is Modesta.
It's an overexposed picture, Dave. You have to compensate for that.
Oh, well,
No,
might
it's
be,
not.
might
This
be
is true
then.
color.
Racial identification is
Yes,
so
Rusula, yeah.
difficult from morphology.
Can you blow up on the cap?
Yes.
Something.
Okay.
Could be, that looks really pale, overly pale for Modesta, though. Modesta has a bloom.
But that looks like a bloom to me on the edges. That's what I was thinking.
Might be.
I don't know.
Tough one. Very tough one. It's a Rusula. I mean, there are so many mushrooms that I bring home. And then I'm determined to figure out what they are. And I don't get past Rusula. And I even look at microscopy. I look at the spores. I mount them in melters. I look at the whether the warts on the spores form any kind of pattern. And still, I don't always get right down to a species. Rustlers are really hard.
Gotcha.
All right, what do we have here?
Little brown mushroom.
A bolete, same as before.
Yeah, it looks like a bolete.
Same?
Xanthoconium athme.
Yeah, Xanthoconium. Yeah.
Okay. How about this? It looks like a Amanita.
Mm-hmm.
Probably some sort of, rubescent Amanita. Yeah, one of the Amera rubescens species.
There's a section view.
Nice photo. Good photo.
Thank you.
A little bit of rubescence in there. You can see this stipe is hollow.
Mm-hmm.
You can see from this the gill attachment is more or less free. It's just in general, from general principles. It's a good photograph.
Thank you.
Yeah, it's one of the rubescent species. The rod has like, I don't know what, like 5 different.
Yeah, 5 or 6 and
numbered species. I mean, a few of them he has changed from like a number to a name, but I'm not sure which. It's hard to keep up with this stuff. Yeah.
It's not just that, you just cannot tell them apart by looking at them. You have to do DNA.
There's 1 or 2 that seem, there's one that has like a yellow veil.
Yeah.
You can tell that one.
Yeah, Oreosibicula. I know that one.
Oh, okay. That's that one. Okay.
All right.
But
so
not this one.
But that's not this one.
No, this is a different one.
Yeah.
This is taller. It had a little bit, also a different.
Yeah, it's probably the same thing as the last one.
Another
Okay.
rubescence, yeah.
Yeah.
Okay, good. So now here's another one, a very small one on this chippable wood.
Oh boy.
But it's very interesting with the shape that it has this. So it has a little bit of the cap had a cup shape to it. I have a bunch, this came on, I realized that the back of the log that I was looking at also had a lot of them. Let me zoom in on this a little bit. You can see the gills don't attach to the stipe. Let me see if I can, so here's a bunch of them. I believe it is a cedar log, right?
I don't know.
Red pine, maybe, or Scott pine.
Some kind of conifer.
Pitch pine, probably.
Pitch pine, yeah, some kind of.
Wow, I have no idea here. No clue.
Okay.
What do you think, John? John, are you still with us? He's John.
I'm mute. I don't know what that is.
Okay.
Yeah, I'm guessing it's white spored. There are so many of these small saprobic white spored things. It's a very difficult group.
Mushroom, you say they're white poured.
White spored.
Oh, that's what
I'm sorry. Think they are, but you should get a spore print.
Okay, I haven't done
you
one
really
yet.
need to get a spore print on these things,
right.
so you know the color.
Here, what do you think of this polypore? Again, older, but they grow together. There's two of them here growing from the one spot, and here's a section view.
Probably some sort of Tolopolis, I would say.
Tolopolis, or I don't think this is Anthoconium athene.
This looks more like a Tolopolis to me.
More like a Tolopolis alboater type of thing.
Yeah, not dark enough though.
Yeah.
For that.
It's just old. It's discolored. I think it is alboater.
Could be. Okay.
I'm going to type it in.
Okay, thank you. All right, I'm going to go much quicker now. I think we had this one already. This is with the powdery stipe, right?
No, that's a different one.
Oh, it is? Okay.
This one is from section Lepidella.
Mm.
And it could be a longipiece.
Here's the section view.
Amanita longipiece.
Oh, I didn't expect to find that here.
No, it's a Pine Barrens thing.
Okay.
You get that one in the Pine Barrens, huh?
Yep.
All right.
Copying that. All right, next.
Possibly. These Lepidellas are very difficult. I mean, the whole genus is very difficult.
All right, I think that's it. So you guys did a great job with like 1 million mushrooms.
Well, it was only like 4, but there was one we couldn't help you with, but no problem.
Yeah, I appreciate it. All right. Does anybody else want to share anything? I guess that's enough. Have a good night. Thank you so much.
Bye-bye.
Good night.
Night, guys. Thank you.
Good night. See y'all later.
Thank you. Bye.